and bringing us closer to the cure, bringing us ever closer to the cure. Following the presentation, there will be plenty of time for Q&A. And so we encourage you to submit your questions via chat. If we're not able to answer them, we'll kind of gather them all up, and we'll be following up with an email that we'll send to the group with answers to those questions. In addition, as you just heard, we are recording this evening's presentation, and we will make it available in its entirety on our website by the end of this week. And Bob, to your point, I think there is a lot of interest in the topic of this evening's presentation. So let me, without further ado, let me introduce our special guest. Dr. Giacomo Lanzoni is an assistant professor at the Diabetes Research Institute at the University of Miami Miller School of Medicine. For over a decade, Dr. Lanzoni has focused on developing cell-based approaches to cure type 1 diabetes. Dr. Lanzoni earned his master's in science and medical biotechnology with honors from the University of Bologna, Italy in 2006, and went on to receive his PhD in stem cell biology in 2010. Since arriving at the Institute from Italy, Dr. Lanzoni has collaborated with other DRI investigators on several key research efforts, including using stem cells for the treatment of diabetes, studying adult human pancreatic progenitors for beta cell regeneration, and developing novel techniques for targeted immunomodulation to protect insulin-producing islet cells. Dr. Lanzoni is an inventor on multiple patents issued in the United States and in other countries worldwide. His translational and clinical efforts benefit from partnerships with industry, including iTolerance, which you'll be hearing more about later, as well as public companies, to advance new treatments from the lab to clinical practice for the benefit of those who are navigating this disease. Dr. Chris Fraker is a research assistant professor of surgery and cell transplantation at the Diabetes Research Institute, and is a member of the DRI's biomedical and tissue engineering team. Dr. Fraker began his career in diabetes research building fully implantable glucose sensors. In 1997, he moved to South Florida with the goal of working in cure-based research at the DRI under the mentorship of Dr. Camillo Ricordi. While at the DRI, Dr. Fraker was encouraged to and aided in pursuing graduate degrees in biomedical engineering, obtaining his doctorate in 2011. Dr. Fraker, his work is focused on optimizing islet stem cell encapsulation to restore normal glucose levels without the need for immunosuppression. Recently, Dr. Fraker has begun examining the importance of innate immune function in autoimmune pathologies and the existence of precursor cells in mesenchymal stem cells. As someone who has been navigating type 1 diabetes for more than 30 years, Dr. Fraker is committed to finding a cure as quickly as possible. He has been at the DRI for 20 years and is encouraged by the increasingly rapid pace of progress and discovery at the Institute. Thank you both for joining us tonight. And gentlemen, the floor is yours.
Thank you so much, Michael, for this fantastic introduction. And I am sharing my screen now. I hope you all can see it. I would need someone to say yes, if that's the case.
Looks good.
Perfect. Okay. So today, Chris and I are here with you for a very special conversation. And we are part of a working group at the Diabetes Research Institute that is called the DRI, the Diabetes Research Institute Devices and Transplant Working Group. And the goal that we are working on is really to eliminate the need for immunosuppression for islet transplantation and islet cell transplantation. And this working group and the affinity groups at the Diabetes Research Institute are bringing in true breakthrough, and thanks to the collaborative spirit that has been established here through the decades at the Diabetes Research Institute, and with renewed energy with the arrival of the new scientific director, Matthias von Herath. He really brought in these ideas of affinity groups and working groups that forms incubators of ideas and solutions to accelerate the pace of discovery and development of new therapeutics and even hopefully a cure for type 1 diabetes. So there are three working groups that are active at this point at the DRI. There is one focused on immunology, there is one focused on devices and transplant, and one focused on beta cells that are the insulin-producing cells, beta cell robustness and augmentation. So this evening, Chris and I want to give you an overview of what we are doing with our teams in the device and transplant working group. And the overall goal for this group is to develop vascularized devices that function as efficient holding tanks for islets. And in a way, efficient also means that those islets will continue to function for a very long time and treat diabetes efficiently for a very, very long time. So I'm going to go through a portion of the presentation, and I want to discuss with you about the harnessing of the immunomodulatory effect of mesenchymal stem cells for the treatment of type 1 diabetes. And later you will hear from Chris about how we combine our ideas and our technologies. So in individuals, in most of us, pancreatic beta cells function very well to control glycemia. But in those living with type 1 diabetes, the pancreatic islet beta cells are directly affected by a condition. And let's look a bit more closely to these cells. They reside inside the pancreas, which is a glandular organ, and pancreatic islets are scattered in this organ. And they are mini organ. They're really small clusters of cells that are able to interact closely to each other. It's actually very important that they interact closely to each other. And the majority of those cells are pancreatic islet beta cells that produce insulin to control glycemia, to control glucose, and to control glucose metabolism. So when islets are healthy, the beta cells are responsible for a good insulin release. And this is possible because they live in an environment that is maintained and protected by protective immune cells. These immune cells have many names. There is one cell type that we care a lot about. These are called Tregs, or regulatory T cells. But what happens when those cells start to lose their function or become dysfunctional, which is something that we see in individuals at risk of diabetes and even with diabetes? Well, what happens is that the protection wanes. And what can happen is that T cells, that is a type of immune cells that can become hyperactive and autoimmune and aggressive ultimately against the islet cells. And they bring their effector function against islet cells, bringing in inflammation and ultimately killing beta cells. There's another type of cells that I want to introduce. These are called macrophages. These are cells that are responsible for organizing an immune pro-inflammatory environment. They also clean up the bodies of the beta cells, so they contribute ultimately to the killing of beta cells. So when these cells start attacking pancreatic islets, they really wreak havoc because they are aggressive. There's multiple types of aggressive immune cells, to simplify things, but the net result is that we observe a major loss of the pancreatic beta cells in those islets in patients, in subjects living with type 1 diabetes. So the central problem in type 1 diabetes is that there are these aggressive immune cells that attack and destroy the beta cells. So in reality, the core of the problem is actually the aggressive immune cells. Those are the ones that go and attack and wreak havoc of the beta cells. And this is true not only during natural disease progression in type 1 diabetes, but also after islet transplantation. Let's say that we have, if we have islet cells that we can transplant, when we transplant those, we have the problem of these aggressive immune cells that are going after either the same beta cells, which is something we call recurrence of autoimmunity, or in general, the transplanted cells, which we call allogeneic rejection or just rejection of the transplant. So ultimately, we think that to solve this problem, we need to modulate those aggressive immune cells. And in a simple word, we call that immunomodulation. So we believe that to solve this problem, we need immunomodulation. And here, let's divide these cells in these two groups. One, you see it in green, the Tregs are the protective cells. But the problem that we see in type 1 diabetes is that they are dysfunctional. They have an impaired ability to suppress the aggressive cells, right? They counteract the function of aggressive cells, these protective cells. But if they're dysfunctional, they're alone, they cannot do much against all of these aggressive cells. And aggressive cells, actually, there's multiple cell types. There's autoimmune T cells, and there's pro-inflammatory cells, and there's a large number of them. And it's just that the balance becomes shifted towards aggressive cells in the context of type 1 diabetes and in the context of transplant rejection. So can we do something about it? And enters our hero. We are focusing on these cells that we call UCMSC. These are mesenchymal stem cells that we derive from umbilical cord. And these cells are very peculiar and special in our view. So these cells can release something that we call extracellular vesicles, or EVs for simplicity, that have cargoes, that have special loads in them that can change the balance. You see, they can change the balance from a heavily inflamed environment, an autoimmune and aggressive environment, towards a more regulatory environment. And they do so by affecting Tregs, which are these regulatory T cells. They determine an expansion and an enhanced regulatory function. So the net result is more immune tolerance, more suppression of the deleterious responses. And these EVs also act on those macrophages. I'm going to bring back to the inflammatory environment before. So the change of the phenotype or the function of those macrophages that shift toward a more anti-inflammatory type involved in tissue repair. And the action that becomes concerted action against the T cells is one of inhibition and blunted effector function. So ultimately, reduced inflammation, reduced cytotoxicity, which means less deleterious effect against the beta cells. And ultimately, we're shifting towards a more protective environment. You see, now we have a lot of cells that are active in a protective way and hopefully shrinking the function of those aggressive cells. So in a few words, the proposed solution is for us to leverage MSCs to modulate immunity. And here you see it in action in an actual experiment that we run in our laboratory. So let's start with what we have here. We have human islet cells, and we're going to do an analysis of what happens to the function of these pancreatic islets. So islets contain a lot of beta cells, and what they do, they release insulin in response to glucose. And when we culture these islets, when we keep these islets in the presence of a low amount of glucose, they release a limited amount of insulin. But when we bring them to a high level of glucose, they will release a much higher level of insulin, which is what they are supposed to do. And when we bring them again in a low glucose environment.
Hello.
Yeah, hi. Can you hear me? Okay. I'll keep going. If you have questions, maybe we can keep them for later, unless it's a really burning question.
That sounds good. Go ahead, Giacomo.
Okay. So we have this islet function that what you see here is the proper good islet function of insulin release in response to glucose and to glucose elevation. If we inflame these pancreatic islets, well, this inflammation ends up inhibiting insulin release from the islet beta cells. So look at what happens. The level of insulin release appears lower and already substantially lower compared to the control, the black histograms here. When we bring also the T cells, these immune effector aggressive T cells, and we unleash them, they end up killing the beta cells and they blunt insulin release. So you see here in red, very low level of insulin release as a result of inflammation and T cell attack. So these beta cells that you see in red are releasing minimal levels of insulin, and they're not even responding to glucose anymore because there's not many beta cells left to do anything in here, actually. So this is the situation where we are in the context of type 1 diabetes, often. The question is, can we use something like MSCs or the MSC-derived EVs, the special mediators that they release, to prevent this loss of beta cells and the loss of beta cell function? Well, it turns out that yes, it looks like that. So look at what happens with the addition of the exosomes, the MSC-derived exosomes. They actually protect the inflamed islets from the T cell attack, and they preserve a glucose-responsive insulin release. So now those islets are releasing more insulin that we call C-peptide. It's a portion of insulin that we analyze, but they're releasing more insulin when compared to the islet inflamed in the presence of the aggressive T cells. And they're getting closer to what they were originally, like real healthy islets, what you see in black. So that is very promising. This is an in vitro setting, but it's really reminiscent of the immune attack that happens in vivo in animals and in patients. So looking forward, we can generate a lot of this material that can modulate immune function. This is just a few pictures from our laboratories. We have a very advanced facility at the Diabetes Research Institute where we can culture the cells, we can process them, we can obtain a lot of these derivatives you see here, and then we can process these derivatives to generate plenty of material such as these immunomodulatory agents. And on separate studies that are very long-term studies that have been pioneered by our colleagues at the Diabetes Research Institute, we have learned that we can treat type 1 diabetes via transplantation of pancreas, via transplantation, importantly, of pancreatic islets that are purified cells, or today, even with stem cell-derived islets, where we think so, it looks like that from in vivo studies in animals, in models, but these stem cell-derived islets today are in clinical trials, even here at the Diabetes Research Institute. So they are getting much closer to a clinical reality. And we're particularly excited about those because they could enable large-scale manufacturing of these cells and ultimately offering the option of transplanting beta cells to a very large number of patients. Here we're showing that when we transplant human stem cell-derived preparations in mice, we actually obtain islet-like structures. So inside the mouse, we can go and analyze what the transplant looks like. We find that it is composed by human cells. You see here this beautiful staining from Dr. Paris on human mitochondria, and this beautiful staining on insulin, marking true beta cells of human derivation in these clusters. And we have very large clusters of the graft and a large amount of insulin-producing beta-like cells. So we know that we can generate these cells. These are important results that we have obtained in collaboration with a company called iTolerance and another international company that is called KadimaStem, based in Israel. So in these animal models, we study whether the transplantation of stem cell-derived islets can also correct diabetes. And so we analyze that measuring blood glucose in mice over time. So this is blood glucose. 600 is very high. We never want to be there. But here below 200, 150 milligram per deciliter glucose is a good place to be. So with these experiments in animal models, we're showing that we can improve glycemia thanks to the transplantation of human stem cell-derived islets. And of course, we're interested in bringing in these special immunomodulators, these immunomodulatory agents. The one that has been manufactured by iTolerance appears to be safe. And here we're transplanting in along with the islets, and the islets continue to perform their effect of controlling glycemia. And so we know that we can treat islets via this form of transplantation. We can treat type 1 diabetes via this form of transplantation, hopefully soon also using stem cell-derived islets. But today, these forms of transplantation require the recipient to be treated with toxic immunosuppression, which is a problem. Okay, this toxic immunosuppression needs to be given constantly to the recipient for the duration of the transplant, for the duration of the function of the transplanted cells. So that is a real problem. And but we said that these MSCs and their mediators seem to be truly modulating immunity. So what we have described earlier is that we have cells and we have derivatives that can interact with immune cells and stimulate the formation of more protective cells and inhibit the formation of aggressive cells. So we actually can leverage this. We can leverage these immunomodulatory mediators in the form of devices. So think about using the cells, or thinking about using the mediators, or even think about using the molecules tethered to these devices, so that a local environment can be generated that will yield more protective cells and less aggressive cells right at the site of transplantation of the islets. That is the goal. The goal is to bring in, leverage these immunomodulatory mediators and bring in the MSCs, their immunomodulatory mediators, in combination with islet cells to reduce inflammation, to reduce the immune attack, and to affect the local immune cell environment so that it becomes protective and not aggressive towards the beta cells. Can this work? Well, these are some of the results from ongoing experiments in animal models. And so in these animal models, we transplant pancreatic islets from donors to recipient mice. And the mice are diabetic on day 0 that you see up here. And after transplantation, their glycemia becomes well controlled. So you see that the orange line and the green line go from this 400, 500 level of blood glucose, which is very high. It falls down here where it's great. But then what happens in the orange line, which is the pancreatic islets alone, is that we call it rejection. This is the direction of rejection by the host immune system, which means that the cells that we transplant are progressively lost over time, okay? But take a look at what happens with the green when we transplant the cells along with these special further immunomodulators and nanomedicine approaches. We get a much prolonged survival of the transplanted islets and a much improved control of the glycemia. So this appears very promising. The experiments are ongoing, but of course we intend to expand these experiments to larger and larger cohorts of animals. So I'm going to summarize the observations so far, and please remember about these key points as we will hear Chris Fraker's presentation, because the ultimate goal is to combine the various parts together to build an efficient way to protect the transplanted islet cells. So we conclude that we can utilize the mesenchymal stem cells, their derivatives, or even engineered microgels to modulate immunity. And we can inhibit the function of aggressive cells, such as the effector cells. And with these approaches, we can protect islet beta cells from immune attacks, with the goal of prolonging substantially the survival of transplanted islets, and hopefully also of stem cell-derived islets. So this is my part. I am happy to take a couple of questions if someone has any burning question or curiosity.
Dr. Lanzoni, thank you. I'm going to step in and ask you a couple of the questions that have come in before Chris takes over his presentation. First question, do you believe the newly functioning beta cells after transplantation are only generated from newly planted cells functioning?
This is a very good question. So in the context of in vivo studies in animal models, we can actually analyze whether there are newly formed beta cells, whether there are newly formed beta cells in the transplanted, among the cells that we have transplanted, or even in the endogenous pancreas, which is the organ of these diabetic animals. We can tell the differences in many ways because the transplant is far and away from where the pancreas is. We can even detect whether new cells form by marking specifically the formation of the new cells with molecules that integrate only in the new cells. Or we can measure if the new cells are very recent in the form of specific biological accumulations such as BRDU. So we have ways to tell if there is de novo formation of islet beta cells, but we actually know that that is a thing, which is very fascinating. We know that the pancreas in humans, as well as in animals, has potential to regenerate to some extent the insulin-producing beta cells. There are amazing studies.
Hey.
Oh,
I didn't know. I didn't know it was this kind of party. Hi.
Sarah, can you go ahead and mute, please? Sorry.
My son. Yes. Look at you.
Thank you, Sarah.
Okay, thank you. So there are amazing results, I was saying, from teams at the Diabetes Research Institute, including the one guided by Dr. Juan Dominguez Bendala and Ricardo Pastori, where the teams are observing live maturation, de novo formation of insulin-producing beta cells. And this is fascinating. The progenitors appear to be there in the pancreas, but the problem is that there is an autoimmune attack in the case of normal disease development, or there can be an immune attack against the transplanted cells. So no matter what those beta cells, where those beta cells come from, we want to protect them. We need to protect them in some intelligent way. Thank you for the question.
We have a number of other questions, but why don't we go ahead and turn to Chris at this point? And then please, everybody, continue to submit your questions via chat, and we will answer as many as possible this evening. And those that we don't get to, as Michael said earlier, we will address and send back to you following the meeting. Thank you. Go ahead, Chris.
Thanks very much, everybody, for having me here. It's a great honor. I guess to start, what I always like to do is tell my own personal story about getting diabetes and what motivated me to come to this place. I got it when I was a sophomore in high school. Had all the classic symptoms, lost all the weight really quickly, didn't know what was going on, thought I was on some new water diet because I was drinking a ton of water and had heard that that was good for weight loss. And I was excited because, for track and field, I'd be ready. when I was 18 in my senior year, I was under fairly poor control and wound up at the Joslin Clinic in Boston. And one night while walking the hallways, I saw this mother, and she was holding a baby that was maybe 9 months old, maybe 10 months old, the same age as my little sister at that time. And she was giving the baby a shot of insulin, the first shot of insulin the baby had had. And the baby was screaming and looking at the mother like, you know, why are you doing this to me? And the mother was also crying. And at that very moment, I mean, I can honestly say that I knew that I was going to dedicate my life to diabetes research, you know, to help not only myself, right? And then I learned later on in my life that two of my children are at high risk for it as well, having the markers, but, you know, also to help something like that. I mean, it just made me, it still moves me to this day, seeing what that mother went through and what had happened. And I mean, I feel a real kinship with everybody here, and, you know, knowing you have loved ones or you yourself are afflicted with diabetes. We can't do this without you. It's a team effort. I'm glad that you're here to see what we're doing. It's important to us. I know one of the biggest things that is always a challenge for me as a patient is over the years of being here, and I've now been here 27 years, which is unbelievable to me. It's half my life. And I hear often from, you know, directors and people saying like, oh yeah, I see that we're going to cure this in 5 years. And it doesn't happen. But the thing is being on the inside, I understand why, both why they say that and why it doesn't happen. And why it doesn't happen is that science takes a long time and you have to be patient. There are small advances, and that's tough for me as the patient. And then why someone like Dr. Ricordi and these people say it is because they are in the game with you. They feel it as passionately as you do, and they want it to be over for all of us, including me, you know. So it's not an easy thing to say. It's like a promise that people want to deliver on. And all I can say is we're getting there. And I wouldn't be anywhere else except for here to do this. And this is why I came here. This is why I stay here. It's important, and I hope you guys all feel as passionately about our work as we do. So with that, I'm going to move on to my talk. Giacomo did a great job introducing this stuff and the kind of collaboration we're doing. As he said, my work is a lot in encapsulation and in methods to address the challenge in cell therapy, delivery, and survival. I mean, our goal is to get away from immunosuppression, because myself as a patient, there is no way I could have immunosuppression. It would reactivate viruses in me and cause problems. So the goal is to do away with that and not have that as a choice any longer. So one of the ways to do that is encapsulation, right? So, but there are problems with encapsulation. There are challenges that have existed for the 40 years that research in encapsulation has gone on. The biggest one is lack of oxygen, right? When you take these cells out of the body, they get oxygen delivered very easily to them in the body, and suddenly they're wrapped in these little capsules and they're separated from their blood supply, and they're also very active metabolically, so they consume a lot of oxygen. So it's difficult for them. They suffer, and when these cells suffer, they tend to send injury signals. And so in sticking with Giacomo's awesome color choices, I chose the red cell for the immune response as well. And you see what happens is that will recruit immune cells to come. And often what they do is they end up surrounding the capsules and then killing the cells inside. So we thought, and we've been examining the fact that perhaps lowering that signal, like getting the cells in better shape before they go into the capsules, making sure they're in the best condition, that you could sort of dampen that immune response. And that's basically what we've been seeing. So we've been doing that in different ways, trying to make these happy islets or happy stem cell derived islets. So some of the ways that other people have done, this was actually work that started at the DRI and then moved with Dr. Stabler to the University of Florida. So we came up with these oxygen generators that were made out of basically hydrogen peroxide, a solid peroxide that then in the presence of water converts into oxygen, and it releases it very slowly because it's in a polymer or a composition that the water doesn't reach it very easily. So it has to diffuse through this stuff, and then it releases the oxygen very slowly. Another system that's out there are these refillable oxygen tanks, almost like a scuba tank for islets. But for myself, the thought of these is that they aren't ideal because Oxygen generation can also produce dangerous free radicals. These extra devices and materials slow FDA approval of anything. And our goal here is to use stuff and to work with things that we can rapidly get FDA approval. So we try to use materials that are already accepted by the FDA and not add too many things to it to slow the actual approval when we get to the clinic. And then the oxygen reloading device requires an open port through the skin, and that increases the potential for infection. So we want to have something that's fully implantable and safe. So I thought of a way of doing this, and we developed these platform drug-loaded nano-emulsions to precondition transplanted cells to protect them against oxygen deprivation and against radical species and oxygen stresses, right? So what exactly is a nano-emulsion? So if you break apart the word, it pretty much tells you what it is, right? Nano is something that's super small and not visible to the naked eye, and this is how these are. And an emulsion is essentially a mixture of oil and water, or water and oil. And I'm sure many of you from food and cooking and things like that are familiar with emulsions. In fact, there are many delicious emulsions. And I give you an example of a delicious emulsion right here, which is like a vinaigrette dressing. So why nanoemulsions as opposed to other methods of drug delivery? So they have increased availability for the drug and increased delivery of the drug in a much more rapid fashion. They're able also to carry drugs that we typically can't deliver because they don't mix well with water or body fluids. So let me explain the concept behind nanoemulsions a little better and why they're so great. And again, this ties into the food area because you'll understand why the flavor is so much better with an emulsified salad dressing as opposed to just pouring oil and vinegar into your mouth. So if you think of a droplet that would hold the drug, let's say it's 1 milliliter, which is a very small volume. The important things in drug delivery are the surface area for the drug to cross over and the distance that the drug has to travel. So when you look at a nanoemulsion, these particles are much smaller. And in fact, in something that's the size of that droplet above, we can have 1.39 times 10 to the 17th particles in a nanoemulsion in that same volume. That's a huge amount of particles, right? I can't even, it's 139 with 15 zeros after it. And that's equal to 5 football fields of surface for the drug to exchange. So as you can imagine, the distance is really small, the area is really large, so the stuff goes out immediately and in a very homogeneous fashion. It goes everywhere and it achieves a concentration very quickly that's therapeutic. This is why salad dressings and emulsions like that taste so good, because they hit all your taste buds really quickly. If you look at the other droplets, it's actually equal to 1/100,000th of a football field. So this is why we target these emulsions and why we think they're a good way to go about this. We've already manufactured some, so we have all the processes in place. Essentially what we do is we use high pressure to turn these components into these droplets. We developed in our lab optical sensors to make production much easier. So in essence, to make these, if you were to do it clinically or to scale, like with a pharmaceutical company, it's very expensive to produce them. One shift of making the drug can cost $1 million for production. And if there's an error during the production, you lose that money. So we developed these sensors to guide our production, to optimize it. And on the left at the bottom of that picture there, you see what a typical emulsion looks like and what the new emulsions look like with our guided production, where we can monitor the process shown in this graph. And we're basically measuring in real time what's going on so we can avoid problems and optimize the particle size and the sterility and everything else in one fell swoop. So this is technology that was all developed in collaboration with a friend of mine and at the DRI. So some of the drugs we've already tested, because we use the same formula emulsion made of all FDA approved components or components that are generally regarded as safe. So all things that would have immediate approval. One is isoflurane, which is an anesthetic, and we've used it as an injectable anesthetic. It works as an induction agent, and it also works to protect islets against cell stresses and hypoxia, which was a surprise that we found out. Another drug is called harmine, which actually comes from ayahuasca, the plant used in different therapies. And it's an agent thought to induce islet growth and proliferation, and it has effects against cell stress as well. Rapamycin, which is an effective immunosuppressive agent in high doses, but in low doses, it's used as an anti-aging medication, and it also is protective against hypoxia and cell stress. And finally, mesalamine, which is a powerful anti-inflammatory from the aspirin family that's used to treat Crohn's and ulcerative colitis. In islets experiments I've done in encapsulated islets, it reduces the inflammation, it improves viability and function of the encapsulated islet cells, and it improves the metabolism and protects them against hypoxia as well. So basically, in the course of these last few years, we've developed a class of substances to test both in vivo and in vitro beforehand to turn on protection mechanisms in the islets or the stem cell derived islets before we implant them into a clinical patient or into animals. And we've seen positive effects in both cases. We haven't gotten to the clinic yet, but we're working towards that. Another area that I'm working in is the need for a standardization of cell therapy dosing and delivery. So when we have encapsulated cells, it's a very crude estimation of what we're transplanting. Even on the clinical level, like islet transplant, you're basically taking a sample from a volume of islets, you count them, and then you use math to basically assume how many you're putting into a patient. So we need something better. It's a rough estimate, and it also depends on the dose collection and the skill of the operator collecting the dose. So these are things that are very variable. And I was seeing that a lot of my results when I would take encapsulated cells and put them into animal recipients, and I couldn't explain the variability I would see from time to time. So we developed a tool that essentially, using a computer script, it automated all the counting and the analysis of the doses that we're putting in. So this script will actually give me a precise dose. It will give me precise information about all the capsules, the size of the capsule, whether or not they contain islets or stem cells, because one of the biggest problems with the clinical application of encapsulated cells is the extra volume of empty capsules that you get, right? We have to minimize that volume, or otherwise we can't translate it to patients. And then finally, it can be used to calculate delivery of immune modifying drugs as well. So as an example, Giacomo is working with this company where they have capsules that contain an immune modulatory agent, but they want to know what the dose is. We can use this software to calculate that as well. So it has other applications. And just to show some of our data where we took accurate dose counts, and then we see that those dose counts actually match the time to reversal in an animal. So when the animal is no longer diabetic, is really dose dependent, as we can see. So this has been a great tool for my own work, and I'm trying to get other centers to use it if they want and things like that. So in conclusion, my conclusions are essentially that we can improve outcome by preconditioning transplanted cells without adding components to the transplant requiring additional FDA approval. And also importantly, staying away from immunosuppressive agents, right? If we're delivering them there, right there locally at the site, we're not going to have the issue of systemic immunosuppression, which is critical to patients. Finally, we can improve and scale precision dosing with the software so that different pharmaceutical companies that are out there delivering cell therapies now, they're actually looking for the same tools, and we're trying to pass that along to them because they want to know what they're giving as a dose. They want to have a more precise way to figure out what dose patients need, how long they last, so on and so forth. So finally, in conclusion, thank you guys again for your time. This is just a list of our colleagues and all the people involved in our affinity group. Giacomo and I actually are leading the group, but as you can see, it's a really highly collaborative effort. We have Dr. Tomei, Dr. Ricordi, Anna Alvarez, and then we have some graduate students, Chris Lee and Donna Shri Survey, my colleague in my lab, Dr. Veludo, Dr. Karupis, who's down the hall, Dr. Vendrame, and then on the other side, Dr. Schuyler, Dr. Lemos, another graduate student, Leonore, Dr. Abdulreda, Dr. Kenyon, Dr. Matal, Dr. Harani, and Dr. Alejandro, and of course, Giacomo. So we thank you very much, and I'd like to open up the floor for any and all questions.
Thank you, gentlemen. We have a number of questions that have come in, and I'll just start off with the following. Mechanistically, do you know how MSC-derived EVs exert anti-inflammatory effects? Might they be influencing immune cell metabolism? And have you considered investigating the combined effects of MSC-derived EVs with Adonoclecl, Lantidra being the brand name?
This is a great question. Thank you, Bonk, for this. So we are studying aggressively the mechanism of effect of the MSCs and of those EVs. So cells work with a whole lot of pathways, and so the analysis is more complicated than everyone thinks, truly, because the cells are actually able to interact with a multitude of different cell types. And so probably they're sending different messages to different cell types. So we need to decode all of this messaging system, and it's intense. It's a lot of work. So we have figured out something. We observe that there are certain mediators and cargoes that are very peculiar for the activation of regulatory T cells, for example, Tregs, which is great. We are very convinced that we can stimulate a proliferation of those cells with such mediators, but there's so much to unravel there. And but it's very exciting. And we consider combining it with Donisley cells. So this Lantidra product is pretty much transplanted donor-derived islets. This has passed the FDA approval, but it follows pretty much the manufacturing strategy that the whole clinical islet transplantation consortium has utilized. So it's something that was developed by the consortium and really spearheaded by Dr. Ricordi and Dr. Alejandro, the team here at the Diabetes Research Institute, Dr. Schuyler, so many of our mentors and collaborators here. So, of course, we want to combine these immunomodulatory agents with islets. But there is a problem, there is a limitation in the number of donor-derived islets that can be even obtained for transplantation in the United States. We hope that stem cell-derived islets will solve the limitation of donor availability, the limitations that we see with the need of multiple donors. So we are working very hard in the direction of stem cell-derived islets. They're not ready for prime time, but of course, we are working on the combination. Thank you for the question.
Yeah, and actually, Giacomo, I'd like to chime in on one thing you touched on. The other aspect of donor-derived islets is that there's so much variability with the islets. You have issues like the cold ischemia time from when the organ is clamped and taken out of the body, and these other issues with organ transport. The nice thing about the stem cell-derived islets is that they're very consistent and very reproducible, and they're cultured the same way, and you don't have the issues that could damage the islets and transport that often affect the engraftment, not to mention things like serology of the donor and things like that can become an issue and eliminate a pancreas from being used. So that's why as a patient, I'm a big proponent of the stem cell derived islets too, and I'm happy to wait until that happens as opposed to donor islets.
Thank you. Another question here. Chronic administration of rapamycin has been shown to impair glucose tolerance in various preclinical models. Although you have talked about the possibility of a low dose, which is known to be beneficial against aging and cell stress, do you know if long-term use in this setting will not result in an insulin-resistant phenotype?
Yes, I'm assuming that's directed towards the nanomorphin. Yeah, so the differences in that dosing are almost 10,000-fold. So yeah, I was happy to see actually that when we give the low dose rapamycin in my hands, the insulin secretion actually improves relative to control islets that aren't being exposed to the rapamycin. Long term, we definitely need to see that. I mean, essentially what I do now is I'm preconditioning them for 24 hours of exposure, and then I take them out of it. It's not something I give long-term because I'm not using it as an immunosuppressant. It's used to turn on protective genes in the islets, as are all those nanoemulsions that I'm using, with the exception of maybe the mesalamine, which I use as a local anti-inflammatory. And then the idea is once those genes are turned on and they get into the body, you really only need a window of time that's maybe 7 to 14 days to address that initial inflammatory attack and hypoxia. And then the cells become revascularized, or the capsules get a nice vascular bed of blood vessels to deliver nutrients. And that's the goal with that, because absolutely long-term RAPA, even at a low dose, we don't know enough about that and what it would do. But that's a great question also. Thank you.
Another question here. Have you tried doing transplantation after X number of days of fasting, followed by additional fasting days after transplantation?
I can respond for in relation to my studies. No, I've not leveraged fasting. Am I getting the question correctly?
Yes.
Fasting? But it's a good point. We have seen publications that are testing fasting to pretty much precondition the recipient. And it is intelligent in the sense that there would be less stress on the transplanted islets or stem cell-derived islets due to the elevated glycemia, for example. So fasting can help. Fasting is one strategy, but insulin treatment is another, very common strategy. If done correctly in many individuals, they can determine good glycemic control. Then there are some individuals that just do not respond to insulin like most others. And for them, diabetes becomes more brittle, more problematic to handle. Yeah, fasting remains an option, but insulin treatment for good glycemic control makes perfect sense.
Great. And one more question. Most patients are not open to cell therapies or transplants if they require systemic immunosuppression, since diabetes is fairly well controlled with devices. And immunosuppression, as you mentioned, Dr. Lanzoni, is harsh and fraught with complications. The itolerance concept of local immunosuppression only is an exciting alternative, but what evidence supports their claim that the immunosuppression is only local and does not affect other critical immune functions throughout the body?
This is a very fascinating area of research. So the itolerance team actually is composed of a number of investigators and absolutely brilliant minds, from Dr. Garcia, Dr. Haval Shirvan, and a large team of investigators at across really the United States. Esma Yolku, who is a close collaborator. So the experience that they have accumulated is wide. And one of the observations that is remarkable, that is still, needs to be fully really described from a scientific point of view, is that immunomodulation imparted by this material may be right at the site of the transplant. If that's the case, it's actually even hard for the current scientists to understand how that happens, because we assume that the immune system travels around the body and can mobilize and go anywhere. So who knows, maybe we generate tolerance to the graft in a site, and then the tolerance may migrate to somewhere else in the body, those tolerogenic cells. Well, it turns out that maybe that's not the case, but definitely there needs to be more investigation around this. And remarkably, the results from the team that went on to develop itolerance point towards a direction of true localized long-term immunological tolerance, or immunomodulation. We can call it whatever we want. But they have remarkable observations in primates, which are monkeys, very similar to humans. So we'll see that the path is not simple when presenting this to regulatory agencies. but there is great hope in that direction.
Thank you. Thank you both. We'll, as I said earlier, we'll collect any additional questions. We'll leave the lines open for a while, so please submit any questions you have, or you can always submit them to info@drif.org. Really appreciate this. Michael Burton, I'm going to turn it over to you to close. Thank you, gentlemen.
Oh, Michael, you're on mute.
Michael, you're on mute.
Oh, my goodness. Thank you very much. It's been one of those days. I want to thank you again, Giacomo. Thank you, Chris, for joining us. I thought it was a great discussion, a great presentation. And Chris, thank you for sharing your story too. You know, I mean, you bring really just a very unique perspective to your work, and that was really quite a moving story. So I don't think there's any doubt that these are very exciting times in the field, and that the Institute is at the kind of the epicenter of progress. We are doing some extraordinary work, and I am just amazed by the passion, the sense of urgency that the researchers bring to their work. It really is inspiring, and I want to thank everybody for being on the call. So many of you are supporters of ours. So many of you are following very closely the work that we do. And simply put, we cannot do it without you. So we're very grateful for that support, grateful for everything that you do for us, with us. And there's so much good work to do still. As Michael said, we will have this presentation up on our website shortly. We've recorded the evening, and so please take a look, or send it to friends, colleagues, people who you think might be interested. Help us tell our story. You know, we have, I think, an extraordinary story to tell, and we rely on ambassadors like you to help us engage others who will benefit when they come to know who we are and what we do. So again, thank you so much for joining us. Thank you, Giacomo. Thank you, Chris. Have a great evening, everyone, and we'll see you next time.
See you soon.
Take care now. Bye-bye.
Everyone.